Thanks for joining us for this symposium. I'm Bill Rapisi, President and CEO of the Lymphatic Education Research Network. LE&RN's mission is to fight lymphatic diseases through education, research, and advocacy. In order to win a fight, you first have to join it. So we ask, please become a supporting member of LE&RN at lymphaticnetwork.org. And we hope you enjoy today's symposium. Thank you.
Well, then welcome everybody. I hope that everybody can hear me, and I'm excited to share with you our story, our work on lymphatics and the brain, or how the tiny vessels impact brain's health. So I'll just show you my disclosure slide, and we can go from here. So as you probably all know, the brain is the most complex organ in our body, and I didn't say most important, I just said most complex organ in our body. It has, of course, two hemispheres, lots of functional domains, and of course billions of cells and trillions of synapses that make this really amazing organ to work together. So, of course, the basic functional unit of the brain is the neuron. We all know that, and of course, there are blood vessels to supply the neurons with the nutrients and oxygen. But there are many other cells in the brain. Actually, the brain is composed only half cells of the brain are neuron cells, even though we are mostly hearing about these cells. There are also other cells in the brain, and the other half of the cells are collectively called glia, and there are several types of them. So, for example, we have the myelinating glia. Those are cells which are wrapping on top of the neurons, and therefore like plastic on wires, and allow a rapid progression of the information. We also have this very, very major glia cell type, which are called the astrocytes, or the star cells. And they're really the stars of the brain. They regulate so many important processes in the brain, and which we'll talk about them later, in connection to lymphatic function as well. And then, of course, so I talked about astrocytes, and then the last part are actually, they're called microglia because they're small. So the microglia, and they are also unique citizens of the brain because they are actually coming from immune compartments. So those are the resident immune cells of the brain. So if we're looking at these cells in a mouse in which these microglia are labeled, and we're doing this window in the skull, and we can look into the brain, what we see is this microglia here in green, and blood vessels here in red. And as you can see, they're constantly active and very, very, very interesting cells. And of course, their role in many neurological disorders is now coming up, and the cells are gaining a lot of importance. So so right now, when we look at these microglia, we are inside the brain. So let us actually zoom out and see what's happening just outside the brain tissue. So here we see a brain, so microglia are sitting here inside the brain. But just outside of the brain, there is this tissue which is called the meninges, or if you wish, the barriers of the brain, the borders of the brain. It's composed of three layers. This first layer, the pia, which is very close, attached to the brain, the dura attached to skull. In between there is arachnoid matter, and then underneath this arachnoid matter, there is this blue thing here. It's a cerebrospinal fluid. It's a fluid that is produced by particular cells inside the brain, and allows the brain to, if you wish, swim within this fluid. And interestingly enough, this area of meninges is filled with different immune cells, all kinds of immune cells. And so I'm not going to go into details, but it is as immunologically diverse as, for example, maybe a gut or a skin, even though gut and skin are exposed to outside world, whereas meninges is not. So of course, the number of cells is much lower than in the gut or the skin, but the variety of cells is really good. So every tissue in our body, except for probably the brain, has two types of vessels, the blood vessels and the lymphatic vessels. And the lymphatic vessels are serving two functions. Function number one is that when immune cells are coming into the tissue to patrol the tissue and to see if everything is okay, then they go out through lymphatic vasculature and into the periphery. outside of the tissue. The other part of lymphatic function is to remove all the interstitial fluids that the tissues produce. So tissues make interstitial fluid, which would wash those tissues with all the toxins that are produced in the tissue, with all the waste products, and all these will be washed out through the lymphatic vasculature. So imagine, so basically if you compare this to your household, your blood vessels will be the water pipes and lymphatic vessels will be the sewage pipes. So they remove all the sewage and all the trash that is in the tissues. So think about your neighbors going through your trash can on a daily basis or on a weekly basis. So they will know a lot about you and about your household, what you eat, where you buy stuff, etc., etc. So same thing happening with the immune system. This waste which lymphatic vessels are removing from the tissues, they're driving into the command centers, if you want, so-called the lymph nodes. And the lymph nodes immune cells are looking through the waste that comes out of the tissue. And if everything is fine, then everything is okay. But if there is some problem in the tissue, if suddenly we have viral proteins coming or bacterial proteins coming, then immune cells know that there is a problem and they rush into the tissue. So the lymphatic vessels are serving a very, very important function of tissue maintenance. So this is happening in all tissues, except for one, or at least that's how it was believed, is that the brain or the central nervous system, the brain and the spinal cord, the belief or the assumption was that they do not have a lymphatic drainage. Which, if you think about it, is quite surprising given the importance of lymphatic vessel function in every other tissue. So we were very interested with this covering of the meninges, like I told you, this meninges, the covering of the brain. And so really talented postdoc in the lab, Antoine Leveau, was able to take this covering of the brain and make them into a whole mount on the slide, like you see here. And then he labeled for different immune cells. And what he found that, so here we have this major sinus is a major vessel that gets all the blood from the brain before it goes out in the periphery. And so all these labeled cells are sitting on top of the vessel because this is a perfused mouse. But next to the sinus, we found these strings of cells nicely organized within the vascular structures. And so we asked ourselves, what are these tiny vasculature which are not the blood vessels? So could these be lymphatic vessels, even though the lymphatic vessels have never been described, or at least not to our knowledge, in that area of the brain, in the meninges. So we actually applied a couple of lymphatic markers, and we were able to see here in magnification, you can see, so this is the sinus in blue, and in red you see lymphatic vessels going just along the sinus. And most importantly, so these are vascular structures, they carry immune cells inside. And of course, because we can do live imaging, we again can make a hole in the mouse brain and we can image how these vessels are draining a waste from the brain. So here, for example, we're injecting green dye into the bloodstream and red dye into the brain. And what we see is that now red dye is flowing through the blood vasculature, and the red dye is flowing through this lymphatic vessel, suggesting that this lymphatic vessel, even though it sits outside of the brain, it is able to drain waste products from the brain. So after we published our paper, another lab of Kari Italo in Helsinki, in Finland, came up with a very, very similar findings to ours. After these two works, ours and Italo's group, there were a series of papers from fish, which is a very important experimental model. So fish also have lymphatic vessels in the meninges around the brain. And there was a paper from rats. And about a year ago, a lab of Daniel Reich from the NIH, in collaboration with us, published a paper that these vessels are also found in non-human primates and also in humans. And so as you can see here by fMRI, the structure of human meningeal lymphatics, which is very similar to what we see in mice. So the lymphatics exist in all species, they conserved evolutionarily, and they probably are important. And now the question is, what are these tiny vessels doing and how they, if at all, support brain health? But before I go there, I actually would like to share with you some historical information about these vessels, because what we were not aware before is that there was a king of Austria who actually wanted to promote education and training by medical students, and he wanted to establish an exhibit or a collection of human organs and parts of human body. Of course, back then, the only way to do it was to replicate it with wax. And so he recruited a very famous Italian doctor, Paolo Mascagni, who together with his artist, Meningeal Lymphatics, were creating absolutely amazing replicas of human organs and human body parts. And so here's the example of the human brain and the human head. And what you can see here, all this white vasculature here on the surface of the brain, and all these vessels coming from the surface of the brain, based on Mascagni, were supposed to be lymphatic vessels. Actually, I got this image from Don Cukigashki from the Department of Pathology at the Medical University of Vienna, and all of these beautiful models are on display in the Film Museum in Vienna. So the problem, of course, is today we know, based on Danny Reich's data from humans, is that humans don't have that aggressive coverage of lymphatics on the surface of the brain. However, of course, we don't know what was the patient that Mascagni was working on to create those models, and it's very possible that a particular patient, luckily or unluckily, had some mutation that made maybe more lymphatics in that head. But regardless, so people didn't believe this is the case. Mascagni's findings were basically completely ignored, and we don't even know where his body is buried, so he kind of died in infancy. And there was this paper in 2003 in a very, very respected journal for clinician scientists in Lancet in 2003 that summarized this whole idea. And so it says here that today there are 867 models on display in the museum. The complexity and accuracy of the models reveal the scientific excellence of their creators and their knowledge of gross anatomy, which is still accurate. However, 4 models stand out for their obvious anatomical misconception. In case number 1 here is lymphatic vessels on the brain surface is presented as obvious anatomical misconception. And it says here that the lymphatic system was the focus of Paolo Mascagni's scientific interest. Mascagni was probably so impressed with the lymphatic system that he saw lymph vessels even where they didn't exist in the brain. The lack of lymphatic circulation in the brain was first described in detail in late 19th century. Of course, if you read this sentence very carefully, one cannot describe a lack of something in detail. But it is very often that initial results are being challenged by people who cannot replicate them or just don't believe in the data, and then very often the initial findings, despite of them being very robust and very true, are dying, or in this case, have been ignored for many, many years. But luckily, I was reached out recently by a very interesting person, a scientist, neurologist, and historian, Stefano Sandrone. And he actually found the original writings of Mascagni, translated them from Latin to English, and we just published this paper that came out last week in Nature Medicine, which discusses the whole history of brain lymphatic system, and actually gives the Paolo Mascagni a tribute for his absolutely beautiful initial description of the system. So now we can go back, and again, when the history is now fixed, and we can now go back and start asking the questions of what these vessels are doing and how they are affecting or impacting brain health. So we are back to our diagram here, and so the lymphatic vessels are sitting here in this area of the meninges, in the dura, and they have access to the CSF, so they are able to get molecules from the CSF. And the question is, how does CSF get molecules from within the brain? And as you can see here, CSF goes into this so blood vessel goes in, and the CSF also dives in. And so if we're looking at the schematics that the group of Mike and Idegaard put together several years ago, what they suggest here is that this blue is again CSF, so fluid. It drives into this periarterial space. And then because arteries are pulsating, this fluid is being pushed out into the tissue of the brain, washes this tissue. So here in the example in purple are example of amyloid beta, which are the building blocks of plaques in Alzheimer's. And this flow is then being reabsorbed at the perivenous side and is then being back removed into the circulation. So what we proposed is that, and then Michael Nidegard called the system the lymphatic system, because she says that there is no lymphatic vessels going into the brain, but the system is functioning as if it is lymphatic, because the water goes through the channels and then is being pushed out. And the cells that are allowing this fluid to go through and to come here through, called the Glia cells, and therefore the name is lymphatic. But what we're showing is that water goes in, washes the brain, and comes out, but the macromolecules, all the waste, if you wish, the solid waste, right, molecular waste, it all goes through these lymphatic vessels. The water, probably most of it goes still through the sinus, but the molecules and the macromolecules, and even probably amyloid beta aggregates, will all be removed through lymphatic vasculature. So the question we asked ourselves, if lymphatic vessels are not functioning and they cannot remove the waste, could it also impair this whole water flow through the brain? So will actually the brain cleansing, or the brain wash, if you wish, will it also be impaired if lymphatic vessels are dysfunctional? So how do you measure brain cleansing or brain wash? And the way to measure it is you can take a fluorescent molecule, put this molecule into the CSF, this fluid which surrounds the brain, and then see how this molecule slowly gets into the brain and then gets out of the brain. So if we do it at different time points, we can see how this red molecule slowly gets in, takes over a lot of space in the brain, and then slowly is being removed. So this is, if you wish, the brain wash, the brain cleansing, or the diffusion through the brain. So, and now we're asking a very simple question: are lymphatic vessels playing, even though they're sitting outside of the brain, are they playing a role in the way the brain is being perfused? And so we can now ablate those lymphatic vessels. We can damage them by different ways, and we have 3 ways to damage them. I won't get into details, it's all published. So we're damaging those vessels, and then we're comparing amount of molecule that gets in and out. And you can see this is a control mouse, and this is the mouse without functional lymphatics, and you can see by far less red here than here, which suggests that the diffusion through the brain, or the brain wash, or the brain cleansing, is not as efficient when the lymphatic vessels in the surface of the brain are not functioning. Now, we do know that, for example, in aged mice, and probably also in aged humans, this diffusion through the brain, this brain wash, is not functioning very well. And we asked ourselves, are lymphatic vessels in aged mice, and 20-24 months of aged mice is probably similar to, I would say, maybe 75, 85 years old human. So we asked ourselves, are lymphatic vessels in these animals dysfunctional? So in other words, could it be that this dysfunctional, this bad brain wash or brain cleansing, could it be because of dysfunctional lymphatic vessels? So we did check that, and indeed we're showing that in aged mice, brain lymphatics are functioning very, very poor. So we said, well, we don't know how to fix this directly, but we actually can fix the function of lymphatic vessels, because if we give them their growth factor that normally they see when they are in a young mouse, we can increase their diameter and we can make them drain better. So what we do here, we apply here a gel on top of the skull, and this gel, it's either empty gel or gel with this molecule, which is the growth factor for lymphatic vessels. And after 4 weeks, we are checking the function of lymphatic vessels. And as you can see, old lymphatics are extremely dysfunctioning. But if we give them growth factor, we can actually very well improve their function. And interestingly enough, this improvement of function of meningeal lymphatics correlates with improved diffusion or perfusion, or again, brain cleansing, if you wish, in a simple language, brain cleansing in the old mice. So again, old mice don't get brain cleansing very well, but if we enhance lymphatic function by growth factor, we can improve this process. And now we asked ourselves a very simple question, it's very important. Does this make any impact on brain function? So how do we measure brain function? Well, we can do cognition in mice. We can take a mouse through different cognitive experiments and see. We know that the aged mice are very, very impaired in cognition. So we can see if this process, which is, again, we're targeting a structure outside of brain vasculature, could it improve the way the brain functions? And of course, I won't be telling you all this if the answer was no. So the answer is yes. So here we're taking mid-age mice, which are impaired but not terribly. And we're taking also very old mice, which are quite badly impaired. And what we're showing here is that in both cases, by treatment with this growth factor, we can improve their learning and memory. And the animals are more using this part of the brain called the hippocampus to actually better strategize to find their escape from this maze, and to find the target and to leave the maze. So this is quite exciting because, again, we're targeting peripheral structures and we're impacting brain function. So the question is now, of course, I showed you that these vessels are involved in aging, they're involved in brain function, and the question is, could then we do something about these vessels? Are they playing also a role in diseases like Alzheimer's? Because, of course, as you know, the main risk factor of Alzheimer's is aging. So our proposal is the following. If in normal brain, we have a brain produces waste materials here in green, a water here in blue, CSF, goes into the brain, washes the brain, is removed out, and the lymphatic vessels are removing this waste outside. But an aged animal, when we have impaired meningeal lymphatics, this waste of the brain accumulates, and basically you create sort of a swamp in the brain. And of course, part of this could be amyloid aggregates, and that could potentially contribute. I'm not saying this is the only factor, but it can contribute to development of Alzheimer's pathology. And so if this is, of course, the case, and you have, if you wish, a clogged sink, then taking out water of the sink will not really help. What you need to do is you need to go all the way down to the plumbing and fix the plumbing. And the question is, of course, will fixing the plumbing in Alzheimer's mice do the trick? So, first of all, we asked ourselves, what's happening in Alzheimer's patients? And what we do know is that if we take brains from Alzheimer's patients, we will see, and patients that will have plaques inside the brain will also have plaques anywhere in the meninges. So the borders of the brain are also very, 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 very packed with the plaques. Interestingly enough, however, if we're looking into mouse models of Alzheimer's, then this is a mouse model. You can see all these plaques inside the brain, but the meninges is completely clean. So the problem is, and I will tell you what the problem is that in animal, the way we have animal models now, because we don't want to wait for 2-3 years until mouse naturally ages to generate Alzheimer's pathology. All the animals models that we have, or most animal models that we have for Alzheimer's, they have accelerated Alzheimer's pathology. And so they develop the disease very early in life. So this is an example of a mouse which is 5 months old, if you wish, 30-40-year-old patient, human being. So it's a very, very old young mouse, 5 months old mouse. And it already has all these plaques because of overproduction of amyloid. But meninges is clean because lymphatic vessels are functioning absolutely fine. So by treating this mouse with this growth factor, we actually cannot improve the pathology because lymphatic vessels are already at their maximum capacity. So we said, okay, but are lymphatic vessels, could it be that, because how come that some people develop Alzheimer's at the age of non-familiar Alzheimer's at the age of 80, whereas others at 95 and have absolutely clear mind? So we asked ourselves, could it be that lymphatic function or their dysfunction may be playing a role in development of Alzheimer's? And here we said, okay, if we can ablate those lymphatics, can we make disease progress quicker and develop earlier? So now we're ablating meningeal lymphatics, and what we're looking at amyloid plaques in the meninges. And you can see these are the control mice, and here the dysfunctional lymphatic mouse, and you can see how all this red in the brain represents amyloid plaques, not in the brain, so in the meninges. And then when we're looking into the brain, then we also see in this mouse here, without lymphatic function, without functional lymphatics, we see more plaques, and they also covering more space, which tells us that meningeal lymphatic health is very, very important. And if they are dysfunctioning, then Alzheimer's may develop faster. We do not have yet data to demonstrate that treating these lymphatics with growth factors will alleviate Alzheimer's pathology, but it does improve learning deficits in very, very old mice. So what we're doing now is we're aging some more milder Alzheimer's mouse models to the very old age, and then we'll try to treat them with amyloid, with the growth factor, and see if we can maybe prevent or better off delay the onset of Alzheimer's in these mice. It is also possible that maybe there's lots of drugs which are available today. None of them, unfortunately, are working very efficiently in Alzheimer's patients. But we believe that maybe combination of therapies, one that will improve this flow through the brain and the other, the existing drugs, made together in combination actually generate some benefit for human patients. Remember also what we also showed you, all these plaques in the meninges, they're also not inert. So here in the red is the plaque that we create now in the meninges, and in green are the immune cells which populate the meninges. So they all attack this plaque. And of course, this attack by itself could potentially detrimentally impact brain health. And of course, in mouse models where this doesn't exist, we cannot really model this problem. But now we can create this by ablation of lymphatics, and so we can in more details can study how dysfunctional meningeal immune response could also be playing a role in Alzheimer's pathology. We also sequenced those meningeal lymphatic vessels from young and old mice and found lots of interesting genes which are differentially expressed. And we also sequenced them from Alzheimer's and control mice. And so we have now several interesting gene targets which we're hoping to develop maybe in the future therapeutic agents that will target those molecules in a hope to enhance the function of lymphatics and then prevent deterioration of brain health and delay the onset of Alzheimer's pathology. So what I showed you here, if I summarize in one slide, in a young mouse, and hopefully also true in humans, when lymphatic vessels are perfectly functional, we have this flow of the CSF through the brain parenchyma, removal of these molecules, and their removal from the CNS, from the brain tissue by the lymphatic vasculature. But in the aged brain, we have dysfunctional lymphatics and accumulation of this debris in the tissue. So the hope is that we can enhance the function of meningeal lymphatics in aged mice using VEGF-C or other methods, and then maybe again enhance brain cleansing and improve brain function. So all this wouldn't be possible without many collaborators that I have, which I really treasure their contribution to our work, and of course without a tremendous support of the funding agencies who are very generously supporting us and allowing us to really do all these experiments. And of course, the most important slide will be the slide to feature my lab members, without whom, of course, none of this would have happened. And so here is Antoine Leveau, who was the first to work on lymphatic vessels in the lab. And then all the Alzheimer's work is done by Sandro Damashkira, and he initially also collaborated with Antoine, and now works very closely with Yasmin, and this is a graduate student, Zach, who also works on understanding the function of this lymphatics, and of course many other people without whom this work wouldn't happen. And as you can see, we're a very diverse lab, and we have people from 16 or 17 countries in the lab right now. So thank you for your attention, and I guess I'll be taking questions. Okay, so here I have a question. Why are lymphatic vessels impaired with aging? This is a very good question, and we don't know really why they're impaired with aging. We do know that they are impaired, not only in the brain, actually everywhere in the body. Everything gets impaired with aging, right? So the skin is less elastic, the heart is not working as well, so muscles are deteriorating. So I think that this is the reason why lymphatic vessels or even the regular blood vasculature are also failing with age. Lymphatic vasculature is very gentle, and lymphatic cells are very gentle, and so this may contribute to their oversensitivity. So here's a question: could you cause Alzheimer's if you ablate lymphatics to help with multiple sclerosis? This is so I did not present somebody probably knows about the work that we showed with multiple sclerosis. So what we did with the multiple sclerosis is the following. We showed that in multiple sclerosis, which is a disease of brain inflammation, there actually lymphatic vessels are functioning too well. They're functioning too much. And so they're removing these molecules from the brain and allowing a very efficient activation of the immune cells, which then come and cause inflammatory autoimmune disease in the brain. So in theory, if we are now enhancing lymphatic function in Alzheimer's, we may in theory cause some problems in inflammatory diseases. I mean, of course, this is always the case, right? Everything is well, everything is balanced in our body. So if you do too much one way or the other, you may of course have side effects, which you probably will. So what we're doing right now is, remember, the function in MS and in Alzheimer's are different functions. So in Alzheimer's, it's a drainage of the waste. In MS, it's activation of the immune system. And so what we are thinking is, because of, so we have all these gene sets now, and so what we are very much hoping is that we'll be able to find molecules which will be able to enhance one function of the lymphatic without targeting the other function, and vice versa, impair one function without targeting the other. So for example, if I can prevent immune cells from getting into lymphatics, yet I can enhance their function, then I have no worries about developing Alzheimer's MS pathology when treating Alzheimer's patients. So again, we are very far away from clinic and from treatment, I think. We are still in a very early, 4 years ago, nobody knew nobody remembered that vessels existed, right? So we're at the very, very beginning. We're just starting our journey. It's very exciting, lots of new things, very interesting discoveries. But I think it will take us time until we're really able to manipulate the system. The beauty, of course, it's so much almost outside of the brain, and it's so accessible that if we really can target those vessels and have a brain's benefit, and to prevent or cure disorders, that will be very, very exciting. So here's another question, great presentation, well, thank you. Are people with congenital lymphatic disorders more likely to be susceptible to brain inflammation, Alzheimer's, or has that been studied yet? So, actually, it's very interesting. What I know from actually talking with clinician colleagues, what I know is that people that have a primary lymphedema, they actually exhibit neurological disorders and neurological problems. So there is link there. I think it's understudied, and I'm hoping that people, more and more scientists, will get interested into this. There are already a couple of works from animals with Parkinson's mice. There is some with stroke, and so I think that there will be more and more to come. Fascinating presentation. Thank you. Thank you very much for listening and for your comments. So if there are any other questions, I'll be more than happy to answer. Okay, where I can find more info on those initial studies. So if you just Google, if you Google primary lymphedema neurological disorders, I think you will find information online. And you can also reach out to me. My email is easily found online, and I'll be able to help to send you some of the citations. Again, it's not very well studied, but I think there is enough information there. And so if you talk to clinicians who know it, they probably will be able to tell you something. So here I have a question. Someone with lymphatic malformation in the lower body having malfunction of the lymphatic system in the brain. So this is a good question. No, we don't know, right? So it could be something local in particular tissue, or it could be a genetic mutation which will potentially induce problems everywhere. We don't know. We don't even know what controls the flow of waste into lymphatic vessels. Actually, Zach is now studying this in the lab. So I think there is lots of interesting questions, but I'm not sure if you have a problem in one part of the body, this means that you have a problem in the entire lymphatic system. I don't think anybody looked into it yet. But again, there are of course genes which are involved in lymphatic function, and of course if you have mutation there, then the entire system is being impacted and affected. And here I have a question: are lymphatic vessels impaired in Alzheimer's patients? Oh man, this is a great question, something that I really want to know. And we reached out to collaborate with several groups, one at the NIH, to do more functional studies. It's a little difficult to study those vessels, the function of vessels in humans. So we're just starting interesting collaboration with colleagues at WashU, and we will be actually getting samples from autopsies from healthy and Alzheimer's patients. And we're planning to sequence those lymphatic endothelial cells. And we hope that by looking not on their function, but what they make, on their genes that they express, we'll be able to derive some understanding whether they are dysfunctional in Alzheimer's patients and what their dysfunction is. So this is potentially very interesting. I guess I'm being told that I missed a couple of questions. Okay, I answered this one. So here it says, does someone with primary head and neck lymphedema have increased likelihood of developing cognitive issues as they are progressing? So actually, I just answered this question, right? based on our findings, this function of lymphatic vessels in the head and neck is very important for brain function. So in mice, we have a very strong correlation there. But again, this is mice, and I think we don't know what's happening with humans. Yeah, I just answered this question. So nobody's asking here, but when I give these talks to clinicians, people ask me, what do I think about, for example, lymphatic massage, cranial lymphatic massage, and so I don't know if by massage you can actually get all the way down to lymphatics because of the skull, but it's not. I think it would be quite interesting to try, and potentially maybe. Really a physical activation of those lymphatic vessels may or may not work. We don't know. So we're looking now in the lab to understand what are the physical forces that can control a lymphatic or improve lymphatic flow in situations where lymphatic vessels are dysfunctional. So if there is a primary lymphedema, there is a primary problem, can we somehow overcome it? Maybe before we have sufficient drugs, can we overcome it by some physical or otherwise targeting them? So we're looking into it, and I don't have an answer yet. We need more scientists, more researchers to actually be involved in this research. That's for sure. And I hope there's more and more coming. And so here somebody is Mark Richardson is sharing some study that I guess we published some time ago. And again, if you just Google my name along with lymphatics, I think you will be able to find a lot of articles. Some are more scientific, and some others are more lay people. Actually, there was I thought it was a nice article. Maybe I'm comparing myself here in Scientific American, August last year. It was called "Immune System: The Seventh Sense." Even though it talks about how immune cells affect the brain, it also has a substantial portion on lymphatic endothelial cells. So here it says, have you or anyone else studied brain tumors and lymphatic involvement? This is a great question, and we are not looking at brain tumors, but there is a lab. I'm not supposed to talk about their findings because this is their work, but I know there is a very, very strong group. And they just, at the closed meeting in January, they presented their very interesting work on lymphatic vessels and brain tumors. So this is very, very exciting. And I will let them finish the study, publish it, and I'm sure that then they will be happy to share and discuss it with everybody. So I don't know if sacral cranial therapy help with the lymphatic function. I don't know. So remember, I'm not a physician. I'm a PhD. I'm a mouse scientist, not a human doctor. I cannot give you really any advice about human health or human disease. And again, I only hope that our findings in mice are exciting enough for our clinician colleagues to pick up on those and actually explore them in humans. Well, I guess if there are no other questions, I thank you all very much for listening, and it was really a pleasure to answering your questions. And please, if you have any other questions, just reach out to me. My email could be found very easily online, and I'll be happy to answer further questions. And then I guess I'll say here goodbye to everybody. And again, thank you for listening.